you, dear colleagues. The talk by Sergey Petoukhov, Consciousness and Genetic Intelligence. The key difference between living and non-living. The creator of quantum mechanics, Jordan and Schrödinger, pointed out the key difference between living bodies and inanimate ones. Inanimate objects are controlled by the average random movement of their millions of particles, and the movement of individual particles is not significant for the whole object. On the contrary, in a living organism, selected genetic molecules have a dictatorial influence on the entire organism. Due to quantum enhancement, see the history of quantum biology. In accordance with this, to reveal the secrets and patterns of living nature, it is necessary to study the universal rules of DNA informatics. Genetic information in DNA is written in the form of long texts of 4 molecular letters or nucleotides: adenine A, cytosine C, guanine G, and thymine T. Science does not know why the alphabet of DNA nucleotides consists of only 4 very simple molecules. But this alphabet of 4 DNA nucleotides is the carrier of a system of binary oppositional characteristics. In particular, 2 of these nucleotides are purines, A and G, having 2 rings in their molecule, and the other 2, C and T, are pyrimidines, having 1 ring. Any single-stranded DNA can be represented as a binary sequence of purines and pyrimidines. Jordan, the author of the first article on quantum biology, argued that the laws of life missed by science are the laws of probability of the quantum world. In search of these overlooked laws of life, I turned to the study of statistical rules in the binary purine-pyrimidine sequences of single-stranded DNAs of genomes of higher and lower organisms from the publicly accessible genetic data bank, GenBank. As a result, universal rules for the statistical organization of genomic DNAs were discovered. Let me explain this. The example of the DNA information sequence of human chromosome number 1, which contains about 250 million nucleotides. The figure shows its fragment from GenBank. By denoting the purines A and G with the symbol 0 and the pyrimidines C and T with the symbol 1, we get a representation of this genomic DNA in the form of a super huge binary number with about 250 million bits. The decimal equivalent of this number goes up to 2 in power 2050 millions. I call these numbers binary genomic numbers, BG numbers. They apparently reflect the laws of quantum biophysics and quantum information science, genetic memory, etc. Mathematical science has not previously worked with system of such huge numbers as far as I know. It turned out that these binary genomic numbers constitute a special subset of the entire set of binary numbers with such multi-million bits. A distinctive feature of BG numbers is the presence in their statistical organization of certain patterns of hierarchical, symmetrical, dichotomous and fractal-like types. To detect statistical rules hidden in binary genomic numbers, they were analyzed by the Oyster's method of statistical nesting dolls, or the method of hierarchy of binary statistics, which considers their structure as analogous to the structure of Russian nesting dolls. This method includes the following stages of analysis of the percentage composition of the binary genomic number. Firstly, it is represented as a sequence of single characters 0, 1, 1, 1, 0, 0, etc. And its percentages are counted, %0 and %1. Then it is represented as a sequence of duplets 0, 1, 1, 1, 0, 0, etc. And percentages are calculated for each type of binary duplets 0, 0, 0, 1, etc. Then similarly, the same binary sequence represented as a sequence of triplets, tetraplets, pentaplets, counting each time percentages of corresponding types of n-plets. The binary genomic number is represented as a multi-layer number, each layer of which is written in the alphabet of the corresponding dyadic group of n-bit numbers. As a result, we obtain a table of probabilities for each type of n-plets in the corresponding n-plet layer. For example, for the binary genomic number of purines pyrimidines in the DNA of human chromosome number 1, the following table was obtained. Initially, there is no reason to believe that there are known trivial connections between families of percentages of n-plets for different n, but they exist. The figure shows the mirror symmetry of the probability distribution in the studied families of binary duplets, triplets, tetraplets, and pentaplets. Red indicates the percentage of n-plets starting with 0 and blue with 1. The interchange 0, 1 produces pairs of complementary n-plets of equal probability. Along with this, in the multi-layer statistics of this binary genomic number, there is a highly accurate dichotomous relationship between the percentages of n-plets of purines and pyrimidines from families with different values of n, for the studied values of n 1, 2, 3, 4, 5. The percentage of any n-plet is practically equal to the sum of the percentages of 2 n plus 1 plets that differ from it by the presence of suffixes 0 and 1. The accuracy of equality is up to a fourth or 5 decimal place. This phenomenological connection is shown here in the form of dichotomous diagram, as well as a corresponding fractal dichotomous tree of percentages, in which each node is the start of its own fractal dichotomous tree of probabilities. These patterns of dichotomies in the statistical organization of BG numbers is valid for all genomic DNAs already studied in our laboratory. All 24 kinds of human chromosomes, all chromosomes of Drosophila, mouse, worm, many plants, 19 genomes of bacteria and archaea, many extremophiles living in extreme conditions, for example, radiation with levels 1,000 times higher than lethal for humans. So Jordan's prediction about the existence of laws of life missed by science, which are the laws of probabilities of the quantum world, is confirmed. Similarly, Schrödinger in the book What is Life stated, new laws to be expected in the organism. What I wish to make clear in this last chapter is, in short, that from all we have learned about the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to the ordinary laws of physics. The reported general genomic rules of probabilities are candidates for the role of such laws. Dichotomies in genetically inherited bodies are well known. Two hemispheres of the brain, dichotomous branching in plants, vessels, and neurons, bronchial trees of human lungs, mitosis of somatic cells, etc., etc. But in genomic DNAs, in contrast to bodily configurations, we encounter a fundamentally different type of dichotomy, dichotomies of probabilistic characteristics in DNA's information sequences. Yes, vast dichotomous fractal networks of genomic DNA's probabilities are the soil from which living bodies and genetic intelligence grow. The material structures of living bodies don't arise out of nowhere, but have structural prototypes in a regular system of genomic probabilities. Let us recall that genetics as a science began with Mendel's discovery of the statistical rules of inheritance of traits. Many processes in living bodies are stochastic, because individual molecules interact in stochastic ways in cells. The discovery of universal stochastic dichotomies in genomes allows us to understand in a new way the phenomena of dichotomies in the inherited design of biological bodies. For example, why over billions of years of life on Earth, is it typical for bacteria and prokaryotes in general to reproduce by dichotomously dividing the body into two halves? What structural and energetic foundations does this eternal dichotomous phenomenon of bacterial reproduction rely on? Which is accompanied by the most complex process of dichotomous separation of all dichotomously organized genetic information, along with the accompanying multi-species protein and nuclein assemblies of the bacterial cell. The following answer is possible. There is a world of families of probabilities, hidden from direct perception, structures on the basis of binary oppositions, like yin-yang. It is in the image and likeness of the binary organized families of probabilities of this multi-layer world that genetically inherited bodies are built. Figuratively speaking, our bodies are like clothes, put on these binary structured families of probabilities, which act as prototypes of biological structures. To a certain extent, this is similar to the situation with the invisible man from the normal wealth, whose invisible figure appeared only when he was wearing clothes. It is also reminiscent of the ideas of the manifest and unmanifest worlds, and Plato's famous allegory about the world of ideas and the shadows on the wall of a cave, by which people living in the cave can judge the true hidden world of ideas. In addition to the purine-pyrimidine opposition described above, in the DNA alphabet of phonucleotides, there are two more types of binary positions: amino-keta, strong and weak hydrogen bonds. Their binary representations define other binary genomic numbers for genomic DNA with a similar multi-million number of bits. I have been established that these binary genomic numbers are also subject to the described rules of dichotomy or twinning of the probabilities of n-plates, with a similar consideration of BG numbers as multi-layer entities. Accordingly, they are associated with their own fractal probability trees, in which each node is the beginning of its own probability tree. All genomic informatics turns out to be connected to a huge multi-layer web of fractal probability trees. The materials of the report are in my freely accessible publications on this website, this year, World Scientific published my large book, Algebraic Biology, Matrix Genetics, and Genetic Intelligence. Second co-author, Professor Matthew He, from the USA. By genetic intelligence, we understand that part of the intellectual potential of living organisms, which allows, on the basis of genetic information in DNA and RNA molecules, to build, for example, from one fertilized cell, an organism with trillions of cells functioning as a whole. At the same time, in such a structured organism, parental characteristics are reproduced in a multi-channel and noise-resistant manner, despite strong noise and constantly changing nutritional conditions and external influence throughout life. I believe that consciousness can and should be considered as a part of or continuation of genetic intelligence, taking into account the achievements of genetics and algebraic biology. Organisms are endowed with innate intellectual abilities for searching for food, escaping from predators, mating and building web of spiders, honeycombs of bees, complex nest of weaver birds, etc., etc. Even organisms that do not have nerve cells at all are endowed with similar abilities. When studying the problems of consciousness and creating artificial intelligent systems, one should look at living nature for algorithms and universal structure of genetic inheritance of such abilities. Part 2. On inheritance of cyclic processes and the relationship of the genetic code with cyclic gray codes. Studies of the molecular genetic coding system revealed its deep connections with the hierarchical family of cyclic gray codes. I'll tell you briefly about this. A living organism is a huge chorus of genetically inherited cyclic processes, the mutual coordination of which is maintained throughout ontogenesis. For example, our body's inherited proteins are engaged in continuous life-death cycles of their assembling and disassembling into amino acids. For example, the half-life of hormone insulin is 6 or 9 minutes. In other words, genetically inherited parts of our body are constantly dying and being reborn in a cyclical manner. Their famous physiologist Gurlich pointed out that the main problem of biology is maintaining shapes with constant renewal of the substrate. Somatic cells of the body divide cyclically. An important role in this is played by proteins with the characteristic name cyclins, the concentration of which in the cell changes cyclically. The energy costs for these cyclic events are taken from the universal source of energy. For all biochemical processes of all living organisms on Earth, ATP, the lifetime of one ATP molecule in humans is less than 1 minute. During the day, one ATP molecule goes through an average of 2,000 to 3,000 cycles of resynthesis. The human body synthesizes about 40 kilograms of ATP per day. Another illustrative example of cyclic organization in life is the metamorphosis of butterfly, consisting of the stages butterfly, egg, caterpillar, pupa, butterfly. It is characteristic that no one teaches the butterfly how to get out of the pupa and begin to fly, but it gets out and begins to fly due to inherited cyclic movements of its ring, cyclic genetic biomechanics. Everything necessary for this is already available in its genomic code informatics. At the pupal stage, the butterfly does not feed on anything. That is, its atomic content is fixed from the beginning of the formation of the pupae. But due to some genetically determined rearrangements in this ensemble of atoms, a new organism ultimately arises, a butterfly. Such facts suggest that the genetic coding system ensures the inheritance of coded cyclic processes based on some cyclic codes. But in mathematics of information coding, there is a wide variety of types of cyclic codes. What type of cyclic codes and their characteristics corresponds to the structural organization of the genetic coding system and their characteristic statistical rules of information sequences of genomic DNAs of higher and lower organisms. I have obtained evidences for the important role of the family of cyclic gray codes for the modeling and analysis of genetic informatics, including the genetic coding system. Received results allowed me to introduce the following effective model postulate. Living organisms are cyclically coded cyclic entities. See details in this preprint. Thank you very much for your attention.